So, a very good evening to everyone present here. We'll just wait for 2 more minutes for everybody to join and then we'll start the session. बेबी, एक बार वो अनु से पूछना कि उसने जो डाल तो दिया है पर उसने कोलैबोरेटर के लिए तो हमें नहीं भेजा। हाँ, पर कोलैबोरेटर का रिक्वेस्ट तो नहीं आया है। उसको बोलना बीट सब लोग को कोलैबोरेटर भेजे। अच्छा अच्छा, आया है आया है। कर हाँ। हो गया। Okay, so let us start the session now. So, a very good evening to one and all present here. Today, we'll be discussing the questions which were discussed in week 8 class. So, which was a continuation of the Drosophila development. So, this week's question will be related to Drosophila development only. Okay. So, the first question is, so as we do always, that I'll just put the question and then I'll give you 2-3 minutes for you to answer the question and then we'll discuss it. Okay. So, the first question is, which of the following is not a gap G? Empty spiracles, orthodentical, cruple, or bicoid. So I'll just label it so that it becomes easy for you to answer. Okay, the first question, this is the first one. A, B, C, D. Okay, so these are the labeling. Question is, which of the following is not a gap gene? Empty spiracles, orthodentical, Krupal, or bicoid. So I'll give you 2 minutes. Then I'll switch the screen. So meanwhile, you think what can be the answer, and then we'll discuss it, of course. Think what can be the answer. Okay. 1 more minute left, and then we'll switch the screen and we'll discuss the answer. Okay, so I'm switching the screen now. Hmm. Okay, fine. The question is, which of the following is not a gap gene? Empty spiracles, orthodentical, Krupal, or bicoid. So first of all, you should know what is a gap gene. So gap genes are zygotic segmentation genes expressed early in Drosophila development. Okay. The example of gap genes are Krupal, hunchback, giant, nibs, empty spiracles. So these are orthodentical. These are broad. They define the broad regions of the embryo. So they are zygotic segmentation genes. So whenever we talk about this is just a minute. Hmm, hmm. So when we talk about gap genes, so these are zygotic segmentation genes. Zygotic segmentation genes, and these are expressed early in Drosophila development. And other examples are Krupal, hunchback, then your giant, empty spiracles, orthodentical. So empty spiracle is a Gap gene. Orthodontical is a gap gene. Krupal is a gap gene. Is bicoid a gap gene? No. So what is bicoid? Bicoid is a maternal effect gene. It's a maternal effect gene. So now comes a question that what is maternal effect? What is a maternal effect gene? So basically maternal effect genes are transcribed in the mother's ovary and mRNA is deposited into the egg. So it acts before fertilization or very early before zygotic transcription starts, and its function is to set up the broad positional information in the embryo. Okay, for example, anterior, posterior, or dorsoventral axis. And the examples of maternal effect genes are bicoid, nanos. So mutant effect of these maternal effect genes are that entire embryo polarity is disrupted. So basically, these are, if I talk about, we're talking about maternal effect genes. So these are transcribed in the mother's ovary. These are the mother's ovary. Okay. And they act before fertilization. It is very important that they act. This is important that they act before fertilization. And the examples are bicoid is a maternal effect gene, nanos is a maternal effect gene, dorsal is also a maternal effect gene, and caudal is also a maternal effect gene. So I hope this is clear. So the correct answer, which is not a maternal effect gene, is bicoid. Now, what is the difference between maternal effect gene and a gap gene? So if I say that what are the differences between, key difference between gap gene, because why I am telling you all this is because these are important points, like important concepts. Okay, you should know these points. And like, hmm, so these are like important concepts, and they form the very basics of Drosophila development. So whenever we are studying about Drosophila axis development or Drosophila development, these are some of the very basics which you should always know and understand. So that's why this is important, and this becomes important, to have an idea about that what these different concepts of maternal effect and gap gene and etc. means. Okay, so maternal effect genes are basically provided by the mother. It acts earliest and it sets up the gradient. Whereas gap genes, they are activated in the zygote and interpret those gradients and refine into regional domains. So basically, the maternal genes are setting up the gradients which are required for the axis formation. Okay, so these which are provided by the mother, and it set up gradient. Okay, just a minute. Hmm, fine. And they set up the gradient. Whereas gap genes, what they refine, so basically they interpret those gradients. Okay, whatever gradients that have been set by the maternal gene, they are read or interpret those gradients and it refine into regional, it refine to regional domains. Okay, so I hope this is clear to you. Okay, next question. The second question is, select the correct conclusions drawn from the ligature experiment on the mixed smita. Bicoid and nanos are involved one at a time in Drosophila development. I'll just label it as A, B, C, and D. Okay, so A is bicoid and nanos are involved one at a time in Drosophila development. B, there must be two decision points, one at the anterior and one at the posterior of the embryo. C, Fushi Tarazu experiment is determined the gradient of the genes expressed. And D, engrailed was seen only at the posterior end of the embryo. Okay, these are your 4 points. I'll give you 2 minutes to answer the thing. Then I'll switch the screen and we'll discuss the concept. Think what can be the answer for this question. Okay. Think what can be the answer. One more minute left. Okay, let us discuss the answer. So hmm, the question is select the correct conclusions drawn from the ligature experiments on the Mitch Smith. So basically, hmm. So first of all, we need to understand about the experiment. So the ligature experiment in the Mitz-Smithia was done to test how positional information is set up along the anterior posterior axis. Okay, so like the positional information has to be set up. So how the positional information is set up? how is this experiment was done to see how the positional information is set up in the anterior posterior axis या फिर along the anterior posterior axis. Okay, then the findings, what were the major findings of this Smithia experiment was that cutting, that is like ligaturing the embryo showed that both the anterior and posterior ends contribute independent organizing signals. That means basically both the anterior and the posterior ends independently they are contributing in organizing the signals. This was the first major finding. Second was that these two, that because there are two independent organizing signals, so there are two decision points, that is organizer, one at the anterior and one at the posterior. In contrast, Drosophila relies heavily on bicoid at the anterior, while Smithia shows more distributed control. So basically important, the point which is important here is that there are two decision points that that there are two decision points. Okay, one at the anterior and one at the posterior. One is at the anterior, one, okay, one at the anterior, one at the posterior. So now let us try to see the options which are given and try to understand them. So A is bicoid, and so the correct answer is there must be two decision points, one at the anterior and one at the posterior of the embryo. Something that we have just now discussed. Now let us see the other points. Bicoid and nanos are involved one at a time in Drosophila development. Is it true? No, because they act simultaneously. Bicoid helps in anterior polarity and nanos helps in posterior. So they are involved one at a time? No, because not one at a time. They are involved simultaneously. Then there must be two decision points, one at the anterior and one at the posterior of the embryo. This is correct. Then Fushi-Tarazu experiment determined the gradient of the genes expressed. Is it correct? No. Because these are pair rule genes, not gradient genes. Fushi-Tarazu, they determined, it should not be determined, it should be determined. Physiotherapy determine the gradient of the genes expressed. No, they do not determine the gradient of the gene expressed because they are pair rule genes, not gradient genes. Because these are pair rule genes. Next is engrailed was seen only at the posterior end of the embryo. Is it true? Is engrailed seen only at the posterior end of the embryo? No, it is wrong. Because engrailed is a segment polarity gene. It is expressed in stripes along the anterior and posterior axis. That means anterior of each parasegment. Okay, not just posterior. So here it is saying that engrailed was seen only at the posterior of the embryo. No, because engrailed, I'll just write it over here. Engrailed, basically engrailed is a segment polarity gene. Okay, it's a And since it's a segment polarity gene, it is expressed in stripes. It is along the whole anterior posterior axis. I hope this is clear. Okay, next question is, which of the following steps are involved in determining the position of the nano's mRNA? Let me just label it as A, B, C, and D. Okay. Which of the following steps are involved in determining the position of the nano's mRNA? A. Initially, the nerve cells deposit nano's mRNA at the interior end of the oocyte, which diffuses into the rest of the oocyte cytoplasm. Then the nano's mRNA is carried to the posterior end on motor proteins like biconic transport. C is at the posterior, Oscar anchors and thus concentrate nano's mRNA. And D, Oscar dissociates nano's mRNA from the SMOC complex at the posterior end. What is the correct? What are the correct statements? Tell, I am giving you 2 minutes. Think. Just a minute. Think what can be the answer. You have 1 more minute. Thirty seconds more. Okay. Let us discuss this question now. So the question is, which of the following steps are involved in determining the position of the nano's mRNA? Now, let us try to understand the correct answer. These are the accepted answers. First, initially the nerve cells deposit nano's mRNAs at the anterior end of the oocyte, which diffuses into the rest of the oocyte cytoplasm. Second, at the posterior, Oscar anchors and thus concentrates nano's mRNA. And third, Oscar dissociates nano's mRNA from the SMOC complex at the posterior end. Okay, so first of all, we should need to know that how nanos mRNA is positioned in the Drosophila. So the first is deposition. That is, nanos mRNA is provided by the nerve cells and it enters the oocyte. So the first thing that happens is deposition. Okay, nanos mRNA is provided by the nerve cell and then it enters the oocyte. After entering the oocyte, what happens? Transport. So what in transport? It is moved to the posterior on microtubules using motor proteins, not bicoid transport. Okay, that's a different pathway. So transport means it is moved to the posterior on microtubules. Then anchoring. What is anchoring? Means at the posterior, the Oscar protein, it organizes the pole plasm and it anchors the nanos mRNA. So nanos mRNA is anchored at the posterior by the Oscar protein. So here, then regulation. What is regulation? SMOG binds nanos mRNA and represses its translation elsewhere. Only Only at the posterior it does the Oscar, plus other factors relieve this repression. So nanos is translated only at the posterior. This is regulation. So what happens in regulation? SMOG, it binds the nanos mRNA and then represses its translation elsewhere. So only at the posterior does Oscar and other factors which relieves the repression. So nanos is translated only by the posterior. So now let us look at the options. Initially the nerve cells deposit nanos mRNA at the interior of the oocyte, which is diffuses to the rest of the cytoplasm. Yes, it is correct. Then the nanos mRNA is carried to the posterior end on motor proteins like bicoid and transport. Is it correct? Is it true? Tell me. No, it is wrong. Nanos is transported on the microtubules by dynein and kinesin, but not like bicoid transport. Okay, bicoid localizes via different anterior anchoring system. So this is wrong. Then at the posterior, Oscar anchors and thus concentrates nanos mRNA. It is correct? Yes, it is correct. Oscar is essential for nanos translocation at the posterior pole. Then Oscar dissociates nanos mRNA from SMOG complex at the posterior end. Is it correct? Yes, it is also a correct statement. SMOG represses nanos elsewhere at the posterior and Oscar what it do? It relieves this repression which allows the translation. Is it clear to everybody? So, what bicoid actually does, it activates as a transcription factor. It activates what? So, just I'll write it over here again. Bicoid. See, these are very important points. So, I'll appreciate that if you are listening it carefully and you are trying to understand these concepts. Okay, these are some of the very important points that need to be taken care of, not just for your competitive examinations point of say also. I mean, not just for your this examination, but competitive examinations point of say as well. Hmm. Okay. So, what bicoid actually does, it activates as a transcription factor. Writing it over here. It activates hunchback. Okay, which is involved in zygotic transcription at the interior. Then giant, which is responsible for anterior domain. Then nerves. It can be activated at low bicoid levels in mid embryo but repressed at high levels in the interior. So it is not a pure activated case, it is activated in certain regions. And then these are the ones which is activated, but it is active nerves is activated in certain regions. Okay. It is not a pure activated case, it is activated in certain regions. Fine. Then it represses caudal. Caudal is repressed. So basically, it is therefore it is present. It is high only at the posterior. Okay. So I hope this is clear to you. Fine. Let us come to the next question. Next question says, which of the following is or are not activated by bicoid? Lot of this has already been discussed. Okay. So which of the following is not activated by bicoid? Hunchback, caudal, nerves, and giant. What is the correct answer? Which of the following transcription factors is not activated by bicoid? Hunchback, caudal, nerves, and giant. Think an answer. I'll give you 2 minutes. Think. Think what can be the answer. Okay, let us discuss it now. Fine. So the question is, which of the following is not activated by the bicoid? Okay. So, hunchback, is it activated by bicoid? Hmm? So correct answer is caudal. Hunchback, is it activated by bicoid? Yes. Caudal, is it activated by bicoid? Yes or no? Is it activated or it is repressed? Caudal. Caudal is repressed. Okay. Nerves. Is nerves activated? Yes, it can be activated at low bicoid concentrations. And giant, of course, is activated. So nerves, it can be activated at, I'm sorry for the noise that's coming in. I think there is some function going on outside. That's why they are playing these instruments and all. Really sorry for that. Okay, so I hope this is clear. Okay, now this is a important and a good question. See, select the option with correctly labeled gradients. Okay, so there are two lines. So there is x-axis and there is y-axis. X-axis may you have Y-axis में you have the concentration. X-axis basically determines the region starting from the interior to the posterior. And you have three lines. One line is of green color, the other one is of red color, the other one is of black color. You have labeling A, B, C, D. You have to tell which is what. And that means that, for suppose if I take bicoid, so which is A bicoid, because the concentration of which factor increases and the interior decreases in the posterior, that you have to tell. So bicoid is basically more at the anterior. So C should be okay. So you just try to answer this. I'm not going to tell you right now. Okay. You first try to understand and then you try to do. Although it's actually easy only. Okay. So I'll give you 3 minutes for this question, but it's easy question. See, if you're willing or if you're planning to give your CSIR net examinations, then the third part of the CSIR net, the C section, they ask these kinds of questions. Okay. So they ask a question related, they ask you these conceptual questions. So there are two things. Whenever you are studying any topic, there are two things. First thing is that you should know the topic. You should understand the topic. First is understanding the concept, understanding the topic, and the second thing is implementing the concept of the topic that you have understood. So there are two things. So you should know how to implement the topic or implement the concept that you have just understood or that you have learned. So these kinds of questions helps you to conceptualize whatever you have learned and then try to implement it in a certain situation, a certain way. Okay, so this is how section C of your CSIR examination works. So if you are good in solving such questions, although this is comparatively an easier one, but if you're good in solving such question, it is very helpful for you if you're going to sit for your CSIR examinations. Okay. So anyways, first you try to answer this question and then I'll give you 3 minutes because you just have to go through a little bit the table and all that, and then we'll of course discuss the question. Okay, so try to solve this. 'Think, see all the options and see the graphs, the gradient, how it goes. Two more minutes left. One more minute. Okay. So now let us try to understand the question. By quiet. So first of all, let us talk if even if you know. Okay, just a minute. Sound is increasing now. I hope the sound has reduced. Okay. So now to in order to answer this question, we should know the function of, even if we know the function of one or two of the transcription factors, we will be able to answer this question. Let us try to start with bicoid. Bicoid is high at the anterior and it is low at the posterior. Okay. So this is the, if you see it over here, this is the anterior and this is the posterior. So it should be high at the anterior and then it should go down at the posterior. So this is C. So C should be the bicoid. So C is bicoid. Then caudal. Caudal is just the opposite. So what will happen? Caudal would be high at the posterior. Okay. And then it will fall in the anterior. This is what caudal. So it will rise towards the posterior. See, it will rise towards the posterior. And then hunchback. Hunchback shows a broad anterior or anterior mid domain, which is activated by this bicoid. See, this is hunchback. So from this, C is the correct answer. Okay. Even if you know two or three, one or two, you can answer. Bicoid, high at the anterior, low at the posterior. This one. Hunchback is the one which can actually help you to determine. Because hunchback is, see, you can say that why this is not hunchback and this is not bicoid. So you have to understand it shows a broad anterior mid domain. So there is a broad anterior basic domain and then it gets little bit activated and then it goes down. This is the basically the graph of hunchback. This one is hunchback. Okay, so I hope this is easy. So like bicoid and nanos are like this simple graphs. This one is nanos. Okay, fine. This is uh, okay. Now let us come to the next question. Just one thing I wanted to see. See what happens because the students are not very interested in joining the class. This is what happens when students are not interested in joining the class. It's very disappointing. So my system has hanged. Okay. Fine. Now let us come to the next question, which is gap genes are regulated by mutual antagonism among non-adjacent pairs of genes. Which of the following regulates pair rule genes? I'll just label it as A, B, and C so that it becomes easy for you to opt. Hmm. So A is promoter, B is methylation patterns, C histone modifications, or D enhancer modules. So a question is gap genes are regulated by mutual antagonism among non-adjacent pairs of genes. Which of the following regulates the pair rule genes? So means which of the following uh, amongst these regulate the pair rule genes? So gap genes, how gap genes are regulated? Gap genes are regulated by mutual antagonism among non-adjacent pairs of genes. This line is not important. This is just a normal line. The actual question is that amongst which of the following regulates pair rule genes? That is, pair rule genes are regulated by promoter or methylation patterns or histone modifications or enhancer modules. So which among the following it regulates the pair rule genes? This is a typical type of question that comes in section C. First, they will give you a whole paragraph which has no connection with the question, and the question is generally the last line. Anyways, you try to answer this question. I'll give you 2 minutes and then I'll switch the screen. Okay. Think you have 1 more minute to answer. Think. Okay, fine. Okay. I'm trying to let us answer this. Try to understand the question. So, gap genes are regulated by mutual antagonism among non-adjacent pairs of genes. Which of the following regulates the pair rule genes? The correct answer is enhancer modules. Now, let us try to understand the question. Okay. Huh. So basically, gap genes. So gap genes. They indeed regulate each other by mutual repression, that is written, that is not just in genes. Pair rule genes, basically they are controlled by enhancer modules. So what happens, that each pair rule gene has multiple enhancer modules, each responsive to a unique combination of maternal effect plus gap gene products. And this modular control, it generates the characteristic 7 stripes of the pair rule genes. Hmm. So what we can see, that the enhancer modules, what it do here in this case. So basically, these enhancer modules, what it do is that they respond, or they are responsive to a unique combination of maternal effect plus gap gene products, which helps in generating the 7 stripe pattern of pair rule gene expression. Okay, so I hope this is clear. Just a minute. Okay, fine. Next question. Is the following statement true or false? Engrailed gene is activated in the cells that will express hedgehog. High levels of even skipped Fushitarazu and paired transcription factors control this activation. In an embryo deficient in Fushitarazu, only 7 pairs of engrailed are bands of engrailed are seen. Is it true or false? Tell me. 2 minutes. Think. 1 more minute left. Okay. So the statement here it is true or false? Hmm? It is true. Okay. Why? Because engrailed, which is a segment polarity gene, is activated in one row of cells per parasegment. Okay. Its activation requires high level of pair rule gene products. Example, even skipped Fushitarazu pair that defines those rows. And engrailed expressing cells also express hedgehog, which signals to neighboring cells. Okay, so in a Fujitarazu mutant embryo, only 7 strips of engrailed form instead of 14 because every alternate parasegment is missing. Okay, in segment polarity gene model, yeah, okay, that is fine. So is it clear? Is it correct? Fine. Now next question is what receptor binds to hedgehog? Hmm. Fine. The question is what receptor binds to hedgehog ligand in the segment polarity gene model? Okay. What receptor binds to hedgehog ligand in the segment polarity gene model? Think. One more minute left. 30 seconds more. Okay. So the question is, which receptor binds to hedgehog ligand in segment polarity? So the to understand this, in segment polarity gene model, what happens? The hedgehog is secreted from engrailed expressing cells. So its receptors on neighboring cells is patched. So when hedgehog it binds to patch, what happens? Patch inhibition of smooth end is relieved, which allows hedgehog pathway activation and target gene activation. So the correct answer would be patched. Okay. So what happens? See, the hedgehog, it is secreted from the engrailed expressing cells. Okay. And its receptor on neighboring cells is patched. So what happens when hedgehog binds to patch? When hedgehog it patch, patch inhibition of smoothen is relieved. Patch inhibits normally smoothen. But when hedgehog it binds to patch, this is relieved. And then what happens? Hedgehog pathway is activated and gene expression is there. And then there is gene expression. Okay. So I hope this is clear to you. Next question is, what is the consequence of deleting the UBX gene in Drosophila? Let me just label it as A, B, C, D. So, what is the consequence of deleting the UBX gene, ultrabithorax gene in the Drosophila? A. The third thoracic segment is absent. B. The second thoracic segment produces all halters instead of the third. C. The third thoracic segment takes the identity of the second thoracic segment. Or D. Halters in the third thoracic segment are converted to legs. Try to answer this question. Think what can be the answer. 1 minute more. 30 seconds more. Okay. Now let us try to understand this question. The question says, what is the consequence of deleting the ultra-bithorax gene in Drosophila? And your options are the third thoracic segment is absent. The second thoracic segment produces halters instead of the third thoracic segment takes the identity of the second thoracic segment, or D, halters are in the third thoracic segment are converted to the legs. So what you have to understand is that ultrabithorax normally specifies the identity of the third thoracic segment, which it directs development of halters instead of wings. So if UBX, that is ultrabithorax, is deleted, then what happens? T3 loses its unique identity and instead takes on the identity of T2. So as a result, instead of halters, the fly develops a second pair of wings, so that the fly has four instead of the two wings. Okay, the correct answer would be the third thoracic segment takes the identity of the second thoracic segment. So is it clear to everybody what would be the answer? The third thoracic segment takes the identity of the second thoracic segment. So what happens, that this ultrabithorax UBX, it specifies the third thoracic segment. So if UBX is only deleted, so what will happen, that T3, it loses its unique identity and it takes the identity of T2. And as a result, what happens? Fly develop a second pair of wings. Therefore, fly, it develops a second pair of wings. So now fly has, so now instead of two. So this is the correct explanation of this question. Next question is, which of the following is a mechanism for maintaining zygotic Hox gene expression? Okay, just a minute. Okay. Which of the following is a mechanism for maintaining zygotic Hox transcription? Okay. Let me label it as A, B, C, and D. A is polycomb protein mediated suppression. B, bicoid gradient mediated gene regulation. C, chromatin modification based regulation. Or D, trithorax protein based activation. The question is, for the maintenance of zygotic Hox gene expression, which of the following mechanism is followed? Polycomb protein mediated suppression, bicoid gradient mediated gene regulation, or chromatin modification based regulation, or trithorax protein based activation. Okay. One more minute left and then I will discuss. Okay, let us discuss this question. So, the question is, which of the following is a mechanism for maintaining zygotic Hox gene expression? Okay, so first is polycomb protein mediated suppression. Is it correct? Yes, it is correct. So it is correct. So basically, polycomb protein mediated suppression, it ensures that Hox genes stay off in regions where they shouldn't be expressed. So it works via polycomb repressive complexes like PRC1, PRC2, or H3K4 trimethylation marks also. Then bicoid gradient mediated gene regulation, this is incorrect. Chromatin modification based regulation, yes, it is correct. So the umbrella mechanism it follows that which includes histone methylation, acetylation, nucleosome remodeling. And both polycomb and trithorax act through chromatin modifications. Okay. Both trithorax, trithorax protein based activation, yes, this is also correct. So what happens, it keeps Hox genes in the domains where they were first activated, and it works via H3K4 methylation nucleosome remodeling, that is SWI SNF remodeling. It works like that. So the correct answer is polycomb protein mediated suppression, chromatin modification based regulation, and trithorax protein based activation. So just again, based, so what does it do? It ensures that Hox genes they stay in they stay off where in regions where they should not be expressed. Okay, and it works via PRC1 PRC2 complexes, that is polycomb repressive complexes, plus H3K4 H3K27 trimethylation marks. Then chromatin modification based regulation, what it does? This umbrella mechanism includes histone methylation, acetylation, nucleosome remodeling. These things it includes. And both polycomb and trithorax act through chromatin modifications. Then trithorax based activation, it keeps the Hox chain on in the domains where they were first activated, and it works via H3K4 methylation, nucleosome remodeling, that is SWY SNF remodeling. So I hope this is just a minute. Hmm. Yes, so with this we have completed all the question and answers of this week of week 8 lecture. So I hope it was clear to you. If you have any confusion, you can always comment in my YouTube channel, and you can always contact me and ask me. So we'll meet in the next class, in the next week class, and we'll discuss the questions which are related to week 8 class. Okay, so bye then. Okay then. Okay. Okay.